Good morning, viewers. Today we'll be sharing about our project Enzyme HaHaHalia. The group members that made this project possible consist of Eldrin, Jeremy, Shintu, George, Navina, and we were all supervised under Prof. Madya Dr. Muhammad Khalizan. So when we were formulating the idea for this project, we were actually thinking about what kind of activities we can done to actually gauge secondary school students and their interests in the field of science. Then an idea was formulated, an idea that would make the event and activity be memorable and at the same time would want full participation from the students. The activity will be built around a concept that we learn in the very classrooms of secondary school in Malaysia. That concept is the lock and key hypothesis that we've learned in earlier Form 4 and Form 5 stages of biology. So it actually involves substrate and enzyme forming a complex which will cause the substrate to form product, which in turn actually explains how the enzyme mechanism works when the substrate is present. So that brings us to our project Enzyme HaHaHalia, which actually involves using ginger enzyme to actually denature and curt proteins from sources such as milk or soy, depending on what kind of media we'll select later on after testing.
Hi, I'm George, and I'm very excited to introduce to you Project HaHaHalia. Project HaHaHalia is a low-cost, interactive module to teach science to secondary school students. The gist of Project HaHaHalia is to use ginger juice, which contains the enzyme zingaban, and warm milk. When combined, it will form a curding, which results in a delicious pudding. So this is a hands-on approach, which means that the students are able to prepare the fresh ginger enzyme extract themselves, and then warm the milk, and then when they combine it, they are able to observe what happens, which is the curding effect. And finally, our facilitators will provide reasonable explanations based on the scientific concepts of the curding process. Project HaHaHalia was designed to build a safe, low-cost, and interactive hands-on module to demonstrate the enzyme activity of zingaban. In addition, it also helps to identify the optimal ginger concentrations, milk temperatures, incubation time, and other parameters to support consistent curd formation. Finally, this project HaHaHalia is also aims to measure student engagement and the interest in biotechnology and the STEM fields after the demonstration. Project HaHaHalia also aligns to the national STEM priorities to push the science, technology, engineering, and mathematics education in Malaysia. Project HaHaHalia is also highly relevant to the biotechnology industry. So in this demonstration, the use of enzyme zingaban, or it is a protease that helps to break down the milk proteins, resulting in the formation of curds. So by explaining this concept of enzymes and the mechanism of action or the activity of the enzymes, we hope to introduce enzyme activity and the usage or applications of enzymes in industry for biotechnology.
Now, I will explain about our project and the scientific principle behind it. First, why did we choose this project? Many people, especially school students, think biotechnology only happens in laboratories with expensive equipment. However, biotechnology can also be found in our daily lives. We chose this project because it uses simple ingredients, which are ginger and milk, that students can easily find and even prepare in their own kitchen. The procedure is simple, safe, and easy to follow with proper supervision. The final product is a soft ginger milk curd. Since we add a small amount of sugar, it tastes more like a dessert, making it more enjoyable and attractive for students to try. Through this hands-on activity, students can see that biotechnology is not something difficult. It can be simple, fun, and a part of a food we eat every day. Before understanding our project, we first need to know what is an enzyme. An enzyme is a biological catalyst. This means it speeds up chemical reaction without being used up during the process. Enzymes are naturally produced by living organisms, including plants, animals, and microorganisms. As shown in this diagram, the enzyme binds to its substrate, speed up the reaction, and release the products. After the reaction, the enzyme remains unchanged and can continue to catalyze another reaction. In our project, the important enzyme is called zingibane. Ginger naturally contains zingibane, which is a protease enzyme. A protease enzyme specifically breaks down proteins. The main protein found in milk is called casein. Therefore, when ginger juice is mixed with hot milk, the zingibane enzyme acts on a casein protein. This enzyme is the key component that allows ginger milk curd to form naturally. Next, I will explain the scientific principle of how ginger milk curd is formed. The first step is the milk contains a major protein called casein. Next, ginger is crushed and filtered to obtain ginger juice, which contains the active enzyme zingibane. The ginger juice is then added immediately into hot milk. It is important to add the juice while the milk is hot because the enzyme works efficiently under suitable temperatures. Once added, zingibane begins to break down the peptide bonds in the casein protein through a process called proteolysis. As these peptide bonds are broken, the casein proteins lose their original structure and become unstable. The unstable casein molecules then start sticking together and form aggregates. Finally, these aggregates create a soft gel structure, producing a ginger milk curd. This entire natural process is known as milk coagulation, where the enzyme causes milk proteins to coagulate and form curd. Our project is an example of food biotechnology. Food biotechnology refers to the application of biological systems or biological components to produce or improve food products. Our project meets this definition because it uses a natural enzyme from plants, which is zingibane from ginger. Secondly, the enzyme naturally modifies the milk protein, casein, causing it to coagulate. As a result, a completely new food product, which is ginger milk curd, is produced. This process doesn't require any artificial chemicals. Instead, it is a safe, simple, and natural biological process. Through this project, we hope the students can understand that biotechnology is not only used in laboratories, but it is also involved in many foods that we consume every day. That's all from me. Thank you.
So initially, when we were developing this project, we were looking at three different protein sources to actually function as the medium for the ginger milk curd. So we were looking at soy milk, fresh milk, and full cream milk. So soy milk was actually not selected as the source because no curd was formed, and when they are preparing soy milk bean curds, the curding process is actually different and actually does not involve enzymes. The second one, fresh milk, was also not selected because it had low protein content and it was not increasable. This is because fresh milk is actually milk freshly obtained from the cow, and when we attempt to dissolve the milk powder in, it was not easily homogenized, meaning the milk powder wouldn't dissolve properly in fresh milk. So that's why we would not select fresh milk, because we cannot control the protein concentration or protein content inside this milk. Lastly, full cream milk was selected as the medium source for the ginger milk curd. This is because actually in full cream milk from Dutch Lady, it is the Dutch Lady milk powder dissolved in water. So we can actually increase the protein content inside this media for better and smoother curds to form compared to fresh milk and the soy milk, which actually did not successfully get or form the curd that we want. As you can see, from the texture of the milk pudding in our early stages of development, it is a very yogurty and watery texture which does not show a smooth curd. And then as the more you try to mix it, the more liquid it will get, meaning the protein actually did not curd properly. So at later stages of testing, we've managed to actually develop solid curds. And I'll pass it to Eldrin to explain what's going on.
Next, I'll be talking about the challenges that we face during this project, and mainly comes from three main factors, which are temperature, enzyme zingiberine concentration, and the concentration of protein. So the first challenges that we face is the temperature aspect. Too high of a temperature can causes the heat to denature the enzyme zingiberine, while too low of a temperature can cause the zingiberine enzyme activity to decrease, which can lead to no curding at all. Why does this happen? Because we are using electric or stove to heat up the milk, which can cause local hotspot and uneven heating of the milk. Note that the zingiberine optimal temperature is around 70 to 75 degrees Celsius. Any temperature beyond or lower than this range will result in no curding. Next challenges comes from the ginger itself, because the type of ginger can affect the enzyme zingiberine concentration in the ginger. Generally, older ginger will have a higher concentration of active zingiberine enzyme, so this can result in an easier curding process, while a younger ginger will have a lower concentration of active zingiberine enzyme, which can lead to little to none curding at all. So the next challenges come from the protein concentration in the milk. A higher concentration of protein in the milk result in easier curding, a more firm texture, while a lower protein concentration can result in a softer texture, but the downside is that it can lead to little to none curding at all. Why does this occur? Is it because total protein or casein concentration will affect the formation of the dense network of the substrate molecule during the enzymatic activity? So more protein means that more network are formed, which result in a firmer texture. So the solution we came up with for the temperature problem is that we should closely monitor the temperature at all time using a thermometer directly measuring the milk temperature at all time and maintain the range of 70 to 75 degrees Celsius. If goes beyond 75, we should let the milk cool down to the optimal range before pouring it into the ginger extract. Next, we suggest that using a water bath to ensure a more even heating will be easier for the curding process to occur. As for the enzyme concentration, we will prefer to use older ginger because it has higher concentration of zingiberine and result in easier milk curding process. However, if you prefer to use younger ginger, it is still viable. However, you will need a larger volume of ginger extract for the milk curding to occur. This could also result in a slightly more bitter taste of the milk curd. As for the protein concentration, the commercial milk that we use only contains 2.7 grams of protein per 100 ml. To counteract this problem, we should add milk powder to further increase the protein concentration so that the formation of curd can occur more easily. Or you can opt for sheep milk or buffalo milk, which generally have higher protein concentration than regular cow's milk. Thank you.
Hi, I'm Shinto Armuga. My metric number is PS2311027 Now I will explain the final procedure for preparing our ginger milk pudding. First, the ingredients required are 25 grams of milk powder, 50 grams of milk, 20 to 30 grams of fresh ginger juice depending on the age of the ginger, and 10 grams of sugar which can be adjusted according to taste. The first step is to prepare milk mixture by mixing milk powder and milk in a 1:2 ratio until smooth. Next, peel the fresh ginger using a spoon, grate it and filter it through a sieve to obtain fresh ginger juice. Then heat the milk mixture to 75 degrees Celsius. This temperature helps activate the enzyme while preventing the milk from boiling. Immediately after heating, pour the hot milk into the ginger juice without stirring too much. The enzyme begins acting on the milk protein immediately. Finally, leave the mixture undisturbed for about 10 to 15 minutes. During this incubation period, the milk will gradually coagulate, and form a smooth ginger milk pudding. Now I will explain the flow of activities during our workshop. We had 4 stations. Participants begin at station 1 where we introduced our project HaHaHalia. We explained the objective of the project and introduced zingiberine, the natural protease enzyme found in ginger that causes milk coagulation. Next, participants moved to station 2 where they safely peel the ginger using a spoon and grate it. This process helps release the enzyme from the ginger tissue. At station 3, participants extract the ginger juice by filtering the grated ginger through a sieve. This produces the fresh enzyme-rich juice needed for the pudding. Finally, at station 4, participants prepare the milk mixture, heat it to the correct temperature and combine it with the ginger juice. They then observe how enzyme causes the milk to coagulate into ginger milk pudding. This 4-station setup allows participants to understand both the scientific concept and the practical application of enzyme activity. Safety was our top priority throughout the workshop. First, the personal protection. Disposable plastic gloves were provided to all participants to maintain hygiene during food preparation. For heat safety, the hot milk was prepared beforehand by us. Participants were closely supervised whenever handling warm materials to reduce risk of burns. Third, regarding safe equipment handling, participants used spoon instead of knife to peel the ginger. This eliminates the need of sharp tools and made the activity much safer, especially for younger participants. Finally, throughout every station, participants were guided and monitored by us to ensure that each activity was carried out carefully and safely. These safety measures help create a safe, enjoyable and educational learning environment for everyone.
Thank you for your attention, and we will see you at BioSpark 2026. Be the first, be the spark.